the Department of Forestry for the invite. I really appreciate the opportunity to get up in front of a whole department that's just a few floors above where I spend most of my time to present some of the research we're doing in my lab. As Mike alluded to, the research focus of my lab is primarily forest management, wildlife relationships, but we do a lot of other type of work. Relative to the forest management topic, though, I'm really interested in this idea of how structural retention functions for wildlife habitat. Now, what do I mean when I use the term structural retention or the phrase structural retention? I'm talking about things like green trees, dead trees or snags, downed wood, understory vegetation that's intentionally left during silviculture or timber harvest operations. And when you kind of think back on the history of why that stuff was left, it has its roots in riparian or water quality protection, right? And then aesthetics. Well, more recently, the benefits to wildlife have kind of been recognized, and that's what really launched my interest into this topic. And when I kind of reflect on why I'm so enamored with this particular topic, it's no big secret, it's a well-known fact that if you increase complexity, it usually has a positive biodiversity effect, right? That's a pretty well-established tenet in conservation biology. However, as I kind of looked at the literature, I felt that industrial forest managed lands were being discounted for their contributions to biodiversity at large scales. And I had the hypothesis that even with just subtle tweaks to our intensive management silviculture or harvest operations, we could actually get a substantial bang for our buck for wildlife, for example. And that was really one of the premises that drove how I built my program here at MSU. Of course, as many of you are aware, best management practices and forest practices regulations oftentimes require the retention of some of these practices. So that's another interest. And then, of course, a lot of you are aware with both SFI and FSC forest certification standards that actually require the consideration of these elements. So that's my spiel in front of funding agencies when I'm trying to beg them for the next years of funding. You demonstrate your importance, why this matters to them. But it's really what's driven my passion for the last 13 years. I'm always quick to point out, though, that I fully recognize that when I ask a forester to leave a green tree or leave a snag, it comes at potentially pretty substantial costs, right? There's potentially lost opportunity cost. You might lose growing space, for example. Or maybe the trees next to a retention patch aren't growing as fast as the trees out in the middle of a clear cut. So I'm very aware of the potential costs that are associated with doing things like this. And I think that, I guess I would attribute my awareness to that factor as maybe one of the keys is how I've been so successful to build that program. Having that in the back of my mind when I'm interacting with forest planners and foresters that got to implement this stuff on the ground has really helped me develop what I think are practical solutions that give us wildlife and productive forests simultaneously. I guess the other, before I jump to my program, the other thing that I was always struggling with is, once I kind of convinced the company I worked for example, that this kind of stuff was important for wildlife habitat, the foresters or the unit managers would come up to me, okay, how much and where? And you better have a really good reason for why you want it, because we've got all these costs associated with doing it. And I was never fully satisfied with the answer I could give to those questions. And as you'll see in some of the results I present from some of the studies I've been involved with, I really sought to try to answer those really basic questions. How much, where, and why is it important? So the work I've done has really focused on two geographies in the United States. I've done a lot of work since 2005 in the Pacific Northwest, and primarily I'm working in the Douglas fir timber types, and then in Northern California and Southern Oregon, a mixed conifer type that includes some pine species in it. In the lake states, primarily aspen, and then as Mike alluded to, we just started some work in northern hardwoods types. I fully recognize that there's other structural retention work that is going on in North America, particularly western and eastern Canada, northeast United States, and the southern United States. And we were actually asked to conduct a review of this topic for forest ecology and management. So rather than dive into the details of that review, I'm going to point you to that reference if you're interested in reading more about some of these other geographies. So today I'm just going to focus on where I conducted my work, Pacific Northwest and the Lake States. As I kind of was preparing this talk and I was reflecting on, what's the take-home message? What really matters when it comes to how this structure, or how these structures function to wildlife in intensively managed forests? I came up with a bulleted list. Obviously, the type of structure, how much, and how it's distributed matters. This is a topic that my lab is specifically focused on. I'm going to provide some results today that kind of show you what I found anyways. The intensity of the disturbance in the surrounding stand is another factor that affects how patches of retention function. The age of the regenerating stand around the retention matters. Landscape context is often ignored, but it actually can be the most important driver depending on which system you're working in. And what I mean by that is, what's the surrounding forest condition or habitat condition in the case of wildlife? What's the broad landscape look like? Is the entire landscape really aggressively, intensively managed? Or are there little patches of management interspersed in a less intensively managed landscape? That has huge ramifications for how structures function. Now, one of my colleagues actually showed a really strong link to the abiotic elements of a particular area, that is an important driver of the species richness or the biodiversity on sites, things like soil productivity and precipitation and those types of things. And then lastly, of course, the species you're studying matters, and then the state variable of interest. And what I mean by that is it matters if you're studying occupancy or abundance or survival. All of those are different states of wildlife populations, right, that we can go out and measure. And the thing we measure matters as we're trying to describe how these structures function. All right, let me tell you a little bit about what we found in my lab. First study I kind of want to walk you through is a study that was funded by the National Council for Air and Stream Improvement. I started this in 2006. So it was one of my first projects that I started when I arrived at MSU. What we were looking at was bird use, and I got a little superscript there because there's a big debate whether it should be called occupancy or use, and it has to do with the assumptions of the modeling, but it's all analyzed in an occupancy modeling framework. One of the hallmarks of the work that I tend to do in my lab is it's usually really covers broad spatial extents, and this project was no exception. You can see we had project sites ranging all the way from just east of Seattle all the way down to Northern California. I had one PhD student on that project, so those of you that are Phoebe smiling, Dan Linden was his name, that tested his ability to pull off a field study. But anyways, we retrospectively identified Douglas fir clear cuts. These were mixed conifer down here. Went in and sampled 20 to 22 stands in any given region. What we did is we would find like a retained patch in those stands and then go count birds around that structure. We'd find a snag in another stand and go count birds and measure how the wildlife was using those structures. Broad spatial extent, decent replication, right, for a field study. Feel pretty good about the inference. So we looked at a couple of different things. Let me kind of orient you to this graph. So each line on this axis represents a different bird species. And these lines represent credible intervals around the coefficient. What you're looking for is if the credible interval overlaps the zero line. If you don't see an overlap, that indicates there's a significant relationship for this variable on songbird occupancy or use. So this is tree count, so basically how big was the retention patch, green tree count. Significant relationship, right? And a lot of you are probably sitting there thinking, well, yeah, no kidding, that's island biogeography theory, right? The bigger the patch, the more species. There's some of that going on here, but I'm going to show you a graphic here in a second that paints a little more. I think we hit a limit as to the species, and knowing where that limit is really important from a management standpoint. I'll show you a graphic in a second. Looked at the quadratic relationship, no significant value there, maybe just for a few species out here on the end. We looked at snag, size of the snag patch, really no significant, consistently significant relationship. And then we even started looking at some of the landscape metrics, like how far the structure was away from the forest edge. Really didn't find a strong signal in the data. So that told us that if we're intensively managing Douglas fir mixed conifer stands out west, we really need to be focusing on the green tree retention, the size of our green tree retention patches, if we want to conserve birds as part of our forest management approach. So we took a little bit closer look at that, and we plotted size of the retention patch by species richness, and that's your classic species area curve, right? I mean, that's what you would expect to see if you were doing island biogeography type work. The bigger the area, the more species you get. But from a management standpoint, what was really interesting to the companies was where that value was. Where do you get the biggest bang from your buck while not adding costs to the company? And we found some pretty clear results, as you can see. This is for upper canopy foragers, gleaners, bark foragers, gleaners, species of regional conservation concern. Really consistent relationship at about 15 trees in a patch. And keep in mind, these are industrial managed forests, so the patch sizes, when I say 15 trees per patch, they're probably pretty consistent, right? They tend to plant those at 400, 500 trees per acre. I don't know, the rotation age stand probably has, what, 100 trees per acre maybe, Doug fir? I don't know for sure. But it's pretty consistent when I say 15 trees in a patch. Pretty consistent area. Don't moan, I'm not going to expect you to see all this, but let me walk you through an interesting result of this. So what I have here, these are just standardized regression values on this axis. Each of my study regions, Central Washington, Southwest Washington, Southern Oregon, Northern California, these are all the bird species that were found in the uncommon group, common group, and abundant group. What I had Dan do here is I was really interested to see if clear cuts with retention were adding anything to the broader landscape. So what we did is we took our data and then we pulled breeding bird survey data from routes that were around our study site. And we did a quick comparison to see, okay, which species are occurring less than we would expect in the broader landscape. So in that case, intensively managed landscape, intensively managed stands could potentially be sinks or not provide habitat for this group. What species are found more than expected in the broader landscape? So in that case, you're making the argument that intensively managed forest stands are actually contributing to regional bird biodiversity. And when you kind of dig into it, you see some really interesting patterns emerge. Like this group right here, Southwest Washington, and these solid boxes are upper canopy nesters, and then the gray shading are primary and secondary cavity nesters. Point of the story is actually in Southwest Washington, you actually see a suite of primary cavity nesters that are actually occurring more often than expected on intensively managed forest lands with structures. So you can kind of see how that type of output could be of a lot of interest, like when you're talking to a forest certification auditor, for example, or you're talking to a public that's pretty critical of forest management, intensive forest management. You say, hey, wait a minute, some of these geographies and conditions have a positive contribution to regional biodiversity. And then there's also the losers, right? And it's important not to just tell one side of the story. I found that out real quick working for industry, because you're quick to be called out on it. The second study I'd like to share with you is a study that we did in Michigan. Started at about the same time, 2005, 2006, somewhere in there. A really cool study looking at aspen clear cuts. And what we did is we identified 222 aspen clear cuts that ranged in age from less than 3 years old to greater than 15 years old, but less than 30 years old. Tried to stratify our sampling in 3 age groups: really young, medium age, older, regenerating aspen. And again, we tried to get a sense for how birds were responding to retention, in this case, in aspen clear cuts. And again, after Clint produced this graphic, I was all excited about it. And then when I'm trying to interpret it for a presentation, I'm like, oh my gosh, what did we do here? I'm going to walk you through it though. Each one of these marks is a different species. In this case, these are mature forest-associated birds. Retention, green tree retention amounts in groups of 3, low, medium, high, low, medium, high, low, medium, high, by the 3 age classes of aspen regeneration. Okay? So, if you have a species that is positively responding to retention amount, you'd expect that relationship, right? Low retention, high retention, as we saw for this red-eyed vireo, we saw a really strong signature. That species seems to be responding to retention positively. And that was consistent across all of the age classes that we looked at. However, you probably picked up on the fact that that was the only species that showed a positive retention effect. The last time I checked, red-eyed vireos weren't necessarily rare in the Michigan landscape. So for the DNR to make the case that we should be doing retention to support the mature forest bird community doesn't really jive, right? And that was consistent regardless of age and across those 5 species that I listed there. There's also concern that if you add retention to early cereal forests, that some of the early cereal specialists might suffer, either from predation by brown-headed cowbirds or raptors picking them off because there's places for raptors to perch. When we looked at a species of high conservation value concern, the golden-winged warbler saw no retention effect. But what you do see is this idea of the surrounding stand age having an effect, right, depending on species. And as you would expect for early cereal species, age of the surrounding stand actually has a main effect on how birds use that clear cut. So again, trying to make the argument that our early cereal bird species of conservation concern are going to suffer because of DNR retention practices I found no evidence to support that statement either. So you might be sitting there thinking, well, why did you find, both looked at bird occupancy or use, real similar stand sizes, real similar bird survey techniques. Why did you find such a strong signal of green tree retention in the Pacific Northwest, but that basically disappeared in the Michigan system? And I think my hypothesis is it has to do with this idea of what's in the matrix. The Pacific Northwest, here's a landscape view of Oregon, and I mean, Weyerhaeuser owns entire drainages out there, right? And they start at one end, and they clear cut, and as soon as those trees are 5 feet tall, 4 feet tall, they cut the next one, they cut the next. I mean, they basically manage, intensively manage at really large scales. So you have a landscape it's intensively managed landscape. Compare that to the Michigan scenario. The DNR manages pretty intensively, right? But as you look at an aerial photo, it's much more patchwork type of a pattern. And my hypothesis is that there's enough older forest condition in between the clear cuts in Michigan that the birds are just temporarily displaced to the neighboring stand. As soon as that stand gets old enough, they just recolonize it. So I've got a couple of students looking at this landscape level, doing some official statistical testing on this hypothesis, and the story's still out on. If they go like the rest of my hypotheses, it's going to totally be irrelevant, right? I also want to say a few words as part of this DNR study. I kind of got sick of working on birds, right? They're highly mobile, they don't stay put in one place, easy to survey. And I was really interested in something that was smaller scale, more dispersal limited, that might be more closely tied to the retention piece of these managed stands. So what we did is we ended up taking a subset of those 222 stands, and we built 45 of these exclosures. That's 3 meters on a side. Flashing is bent in. And what we did is we caught a bunch of these redback salamanders and marked them and put a known number of salamanders in each exclosure. Let them sit for a month, come back, see who we could find. Let them sit for another month, come back. It's like a marked recapture study, right? To get a sense of survival, parent survival probability. So what we looked at, and this is where that I made that comment about state variable matters. So the bird stuff all looked at occupancy and use. When we looked at salamander abundance, just salamander count, we found no relationship to retained canopy cover, that's the black line, or the number of coarse woody debris. No effect on salamander counts. However, if we look at a different state variable, in this case apparent survival from our marked recapture data, we actually see a pretty significant effect size for canopy cover, and then less of an effect size for coarse woody debris. One of the things that these data hammered home for me was, and wildlifers preach on this all the time, kind of the gold standard for wildlife studies is to figure out something that's related to lambda, or the population growth potential, the population growth rate. A lot of times things like occupancy and abundance, they can be weakly correlated to population parameters. But when you start really digging into what I think is an important population level parameter, it can kind of change your inference or how you view the results of your study. So that really hammered home for me the importance of the state variable that you were looking at. So up to this point, I kind of put check marks by all of the state variables that I presented some evidence. And I apologize for some of you, this is probably an annoyingly insufficient amount of detail, because I know how scientists are. They really want to know, well, was your p-value, you know, and all that kind of stuff. So if you want to talk about that, we can do it at the mixer, but I chose to stay pretty high level so I could cover a couple of different projects. So we kind of covered these state variables, and I'm involved right now with a study that's going to help me kind of get a sense of density in addition to some of these other ones. Over the course of my career, my work has kind of evolved from, I call them retrospective studies, you know, where you don't necessarily plan the experimental design, but you go out and you see if you can find 23-year-old clear cuts with really no idea how they got to be 3-year-old clear cuts, but they're kind of your replicates. So you kind of take the hand you're dealt. That's a retrospective study design, kind of a space for time substitution for those of you that might be familiar with that. I really tried to move into manipulative type studies to really start isolating the retention effect, right? So if I've got a tightly controlled experiment, it really lets me hone in on the thing that I'm interested in this case, the retention treatment. So about 3 years ago, I was fortunate enough to get convinced Nikazi to fund a follow-up study out west, where we actually worked with a whole variety of landowners. And just to get you oriented, this is Portland, Oregon, right here, Columbia River, so that's the border between Washington and Oregon. We extend, this study site, the extent goes east of Longview, which is right there, all the way down to kind of like by Corvallis in Oregon. We identified 10 blocks and implemented a randomized block design where we picked 5 stands in each block and randomly applied a treatment, retention treatment to those stands, and then worked with the companies or the landowners on getting those cut the way we wanted them to. Huge effort trying to coordinate that across this sized area and this many landowners, but we were actually able to pull it off. Really exciting design that'll keep giving long after I'm retired, right? I'm looking at the front end of it. My successor is going to have this design to work with into the future. Same with our Heartwood project. Let me give you a sense of the treatments that we were really trying to isolate. Treatment 1 was called our riparian treatment. So standard forest practices, right? You got to protect the water course. So you leave a buffer of uncut trees along the edge of the stream. And what landowners like to do is they like to leave their retention trees that are required under forest practices rules, 2 per acre, right along the riparian zone, because it's easier, right? The helicopter's not going to fly into them, the planting crews can, you know, it's just out of the way. So that's kind of the standard way of doing business, is to pack them all down along the riparian zone. Forest regulators were asking the question, well, we think you should have some upslope retention because it adds wildlife value to these treated areas. So that's the question we were trying to get at with this experimental design. So we took our retention trees, and one of our treatments was to move it all upslope in one big patch. We did a split treatment where we put half of them down around along the riparian zone, half upslope. I get it. We also, there was some research conducted that suggested creating snags was a good way to enhance the value of these retained patches. So what happens here is they'll take the head of the harvester, reach up as high as they can, nip it off, take the top for merchantable, but you leave the 20 to 30 foot, you know, of stem as a potential snag or a future snag. So that was the split retention with created snags. And then dispersing these things all over in patch sizes that were at least 15 trees. Going back to my first study, right, that's the optimal patch to maximize bird diversity and disperse those throughout the landscape. Obviously, the landowners like this one, hate this one. I really changed the focus, kind of like I did with the salamander study, is I'm kind of done with birds, and I apologize to anybody that's researching birds in here, but I think I've run the course with what I can learn about bird occupancy and use with forest management studies. So I moved to organisms that, again, I felt were more closely tied to the treatment. In this case, we're looking at small mammals and then the ground beetle community, or carotid beetle community. Now, when I'm in front of a panel of 20 member companies from the CASI, which is the National Council for Air and Stream, my funding agency, and all the company reps are sitting like you guys are, and I got to convince them to study carotid beetles and mice. That can be a challenge, let me tell you. But fortunately, the companies bought into it and funded the project. This is the case I made, is these are actually very important organisms from an ecosystem function, structure viewpoint. And when you kind of present it in this context, it's a lot easier to get on board and buy into why studying this group of organisms is important. We implemented everything fall of 2012, finished the harvest in summer of 2015, and let me just present you some preliminary results from the beetle and the small mammal work. We used pitfall trapping to capture the carotid beetles, and we had 2 month-long trapping sessions. Just to give you some sense of the magnitude of the data that we're collecting, in 2017 we had 48 species and almost 6,000 individuals that Sean Saltaire, my PhD student, keyed out each one, so good experience for him. Sidebar, in North America there's about 2,000 species of carotid beetles. The beetle expert we work with in Oregon expected us to find about 25 to 30 in the landscapes we worked in. So there appears to be higher species richness than what some of the experts would have projected. But let's face it, there's not a lot of people looking for them, right? So you got to kind of take that with a grain of salt. I also want to explore this idea of functional diversity as part of this study. Species richness is like your most basic measure of diversity that you can use. And this idea of functional diversity, where the diversity of morphology and behavior are really the important drivers of how the ecosystem functions. That's what I want to explore with this beetle data set. And you see one example of the range of functional traits just based on beetle size, right? These are all from our study, adults. There's no young ones in there, all different species of beetle. Even the specialization of the jaws or the mandibles for foraging on different things. So that's how I'm approaching diversity with this study. I think it'll be a really interesting look at it. Give you some sense of the small mammal work. We only visited each stand once, trapped for 4 nights. We did some pilot work that showed that that was enough trapping to get a reasonable estimation of the small mammal community. Consistent trapping effort across all our treatments and our stands. And so far we found 22 species. Obviously we got some oddballs, spotted skunks and things like that really won't probably factor into the analysis. But again, pretty happy with our overall ability to catch a variety of species. This is a risky graphic to show, because the industry guys really like it, but it's only based on 1 year's worth of data. Individual small mammals in a sample, species richness on the y-axis. What the industry liked about this graphic is that our riparian treatment seemed to be the one that supported the greatest species richness of the small mammal community. Now, some of you are probably sitting in your seats saying, but you're probably getting a bump because of the fact that it's connected to a larger riparian zone. Yeah, exactly. So we got to, and I always present that caveat when I present this result, because we got to tease that part out. It's a bigger functional patch, right? Because it's right alongside this big riparian zone. I think that's what's driving that bump. All the other ones, though, you know, pretty consistent. We also sampled some unharvested stands. That's the yellow line. It's kind of buried in the middle here. Just as something to compare to. We really weren't interested in comparing clear cuts to old growth. That's been done a million times. And, you know, big surprise, clear cutting is bad for species that prefer old growth. I didn't need to show that again. But what I was really interested in was, do these rotation age stands, which dominate this landscape, is there something unique in there that we're losing by all this aggressive management? And so far our results are saying no. Actually, they're actually really depauperate, because if you're familiar with those systems out there, it's a closed canopy. There's no sunlight hitting the forest floor. You get sword fern and moss basically on the forest floor. And it actually is a pretty, kind of like a real pine plantation in Michigan. I won't spend a lot of time on this, but this is that density piece that I mentioned I was really interested in. My student is using some spatial mark recapture model. It's a pretty powerful way to estimate density. And the whole point of me showing this slide is that for some species, like Townsend's chipmunks, we saw a real strong effect of retention overall. So retention is orange, the clear cut is gray. Townsend's chipmunks are clearly honing in on the retention in these harvested stands. Really not a strong treatment effect, so this is riparian to dispersed and everything in between. However, when we look at deer mice, year one, we saw a real affinity for the clear cuts. Last year, that completely flipped. Not quite, it's starting to flip. Now, it'll be really interesting to see if the flip becomes even more pronounced this next year. Not really sure what's going on there. We're thinking that it might be a signature of the regenerating stand getting to a point of development where you have less open ground and maybe seed-producing plants or something related to the vegetation condition. And then the exact opposite, creeping voles really showed a response to the clear cuts and not so much the retention patches. However, last year creeping voles started to show a treatment effect. So the point is we've got some really neat data to estimate a treatment effect on density for a whole suite of these species. And again, I think it's a step up from simple occupancy and use type of analyses. Just a few take-home message, and a lot of these are question marks because I think we still got to sort through it. Small mammals appear to be showing a treatment effect. One of the things Sean is going to look at is the effect of retention patch metrics. So distance from edge, how convoluted the patch might be, those types of things. And then also landscape context, this idea of what's around it on the small mammal community. And then for the beetles, there's some early indications that they're more species rich than we previously thought or that were expected. We're seeing what we think is a lot of functional diversity. Too early to tell if we're going to see a treatment effect or if those species are really keying into just on real local, you know, there's a piece of down wood there or something more localized that they're keying in on. So we'll try to tease that out with this next phase of Nakazi research. Now let me end by talking about the Heartwood study. After you introduced me, I was just going to say, any questions?
Sorry about that.
No, that's okay. I'm really excited about the opportunity to work with Mike on another DNR project that we started, 2 or 3 years ago, I guess, DNR Northern Heartwood Study. And apologize for the picture, it's a little dark, but I know a lot of you in this room can relate to the issue that's showing up in this slide. It's all this beach regeneration, right? It's one of the challenges that this forest community type is facing in Michigan, is trying to outcompete really aggressive beech regeneration. Our project is actually taking a broader look at this northern hardwood regeneration issue. Our goal is to try to figure out a way to produce diverse, adequately stocked northern hardwoods. I think one of the things we're struggling with is exactly how we quantify that. Mike provided some really good guidance on what adequately stocked means, but probably the big unknown one for us is, what does that mean? You know, is that 2 oak per acre, 5 oak per acre, 2 white pine per acre? You know, we're still trying to sort out what that might be, and I'm sure it relates to site conditions, right? But the project is also quick to recognize that this is just not a deer herbivory issue. There's a lot of things going on that have gotten the northern hardwood stands of this state into the condition they're currently in. And it's kind of funny because before I even got involved with this project, I serve as an internal forest certification auditor for the DNR. So I'd go to the management units and we'd look at these stands. And I would keep making the argument that I don't think this is sustainable forestry. You know, you're seeing small stuff in the understory, but as Catherine's work showed, it's like 80 years old or 40 years old. You know, it's just not growing. You're not seeing the regeneration. And to be fair to the DNR, the selection silviculture system really isn't viewed as a regeneration type of cut, right? You kind of continuously select, and you assume you're going to get a few recruits to replace the selection. I wasn't seeing that happening. Mike had some data that showed it wasn't happening, and I think that was the impetus, one of the reasons we got this project started. So we recognize deer are undoubtedly a big part of the equation that affect our ability. But we're also quick to recognize that insects and disease, that beech thicket, probably from beech bark disease, right, killing all the parents in that stand, stimulating suckering. There's this management legacy that I just talked about, 40 or 50 years of selection silviculture that has, you could probably argue, hydrated some of these stands. And then, of course, the changing climate is going to have a role on our ability to regenerate this type of forest type. I think our hypothesis, our overall hypotheses going into this is that we can actually do some alternative approaches to silviculture that some would view as more aggressive than typical northern hardwood silviculture to meet this goal. And really our whole design is set up to try to answer that question. Big study again, 141 sites, randomized block design, really nice design again. DNR and private partners got them all cut within 13 months, 14 months. So basically last winter. Huge effort. Again, we got to take our hats off to the DNR for pulling this one off. It's an amazing study design. Has its challenges logistically with field crews, right? And then I know some of you in the audience, and I'm almost done here, are probably interested in the treatments themselves and some of the details of the treatments. This is our standard way of doing business, right? Selection, silviculture with maybe an occasional small gap. We implemented a new treatment called shelter wood, where we tried to retain about 50 BA throughout the stand. We implemented a pretty aggressive seed tree where we tried to retain 6 to 8 diverse seed-bearing trees per acre throughout the stand. And then a large group patch cuts where we really ratcheted up the size of the opening to 1 acre maximum. So randomized those across our treated areas, and then we also had 2 understory treatments. A really aggressive herbicide and scarify treatment, and then what we're calling the leaf treetop treatment, where we asked the contractors, the loggers, that if there was anything bigger than 9 inches, to leave it on the ground intact. So stay off of it with the skidders, keep the volume to it. And the idea with this treatment is that we can create physical barriers that potentially deter deer from accessing the desirable species. The hypothesis with this one is that we can actually overwhelm the site. One of the hypotheses is we can overwhelm the site with browse, and some of our desirable species can make it past the browse zone. So my job on the project, Mike's handling all the silviculture stuff. My job is to kind of get a sense for what the deer are doing on our different treatments. And almost every trail cam photo that we collect, I've got a deer eating one of Mike's sugar maple seedlings. Those doggone deer. But we're using a whole series of remote cameras, really nice experimental design. The challenge here is pre-treatment, we had almost 300,000 photos we had to sort through and photo tag. So that's been our biggest challenge with that part, is trying to manage all the data that are coming in. So we just started that, so you got to stay tuned for some of the results on that. Maybe I can give another Hanover lecture in 4 or 5 years or so, come back. So hopefully I've given you some sense of the work we do in our lab, that I actually have conducted research that hit on most of these things that matter to how structures function in intensively managed systems. And one of the things that sticks with me is Dave Weeman's chief forester out of Northeast Washington used to work for. I'd always ask him, leave me a snake, leave me a patch, leave me a green tree. And he would always ask me, does this stuff do any good? That was always his question. It gets back to this how much, where, why, leave it. And right now, 13 years later, I feel pretty comfortably saying unequivocally yes, that this stuff, if we're thoughtful about how we leave it, can actually have a pretty substantial increase in the wildlife contribution of intensively managed ground. So that's my presentation. Thank you very much.